Thanks so much to Lale for inviting me to this meeting and to give this talk. As Kelly mentioned, we're sort of new to G&E myopathy, but we've been studying carbohydrate biology for a very long time. And so our lab I wear two hats. In one hat, I try to understand at a very basic level the science of how it is that when you put sugars onto proteins and onto cells, how does that change the biology of the cell, how the cells communicate with each other, how they sort of maintain their integrity. And we test that at a number of levels, but particularly at genetic levels. And then with my other hat, I design gene therapies for patients with muscular dystrophies. And I'm going to tell you one very short story about our first gene therapy that involves changing glycosylation of the muscle membrane, which is not G&E gene therapy, but is now in clinical trials, and sort of tell you a little bit about the process that got us through to getting to the clinical trial, and then tell you a little bit about Kelly's doctoral work on G&E myopathy. So first we have to please the lawyers at the hospital. And so I received funds from Sarepta Therapeutics for this gene therapy I'm going to talk about, which is GALGT2 gene therapy. So you just need to be aware that I have a financial conflict of interest in talking about that. And I have to give credit to a very large number of people. It's very easy to get up here and talk about a large body of work like this, but it actually is done by just a gigantic group of collaborators. And so you can see Kelly hasn't aged a day since this picture was taken. So she's deserves all the credit for the G&E work. I'm going to talk about Ray Zhu here in the sporty red jacket, has developed the gene therapy for GALGT2. So my life at Nationwide Children's Hospital and Ohio State University involves collaborations with a consortium of about 25 faculty that all specialize in muscle biology and muscle disease. And a number of those are physicians. So Kevin Flanagan here gets in bold because he's the IND holder on GALGT2 gene therapy. Without him, we couldn't have moved this to a clinical trial, but also Jerry Mendel and Louise Rodino-Claypack were instrumental in that process. And then I'm going to talk a little bit about sialic acid biology, where I've used technologies from Ajit Varki at UC San Diego, which is where I originally started my career. But we have this giant clinical team as well, and this is just to tell you that without a coordinated clinical team like this, you really can't bring these trials into place. So we have a cardiologist, John Cheatham, who did the vascular delivery for this gene therapy. We have physical therapists, Megan and Linda, who do the patient testing. We have additional physicians like Megan Waldrop, who also help out with the trial. We have nurses like Rachel Schrader that are clinical research nurses. We have clinical research coordinators like Federica and Tabitha. We have FDA regulatory specialists like Patricia, and then we have technology transfer agents like Margaret. And the key thing here is that the hospital funds all of these people. They all have PhDs. They all have specialty in gene therapy. And you really need a team like this to move products forward. So I've just learned something here today, which is that you should never give a talk after your student has given a talk, because they're going to upstage you. So Kelly's slides are much prettier than mine, and she's already sort of explained all of this to you, so I'm not going to go into it. But suffice it to say that for adeno-associated virus, making a gene therapy is fairly simple. This is a virus that only has two genes in it normally, a rep and a cap gene. You can take those out, and if you just leave these little red blocks at the end, which are the inverted terminal repeats, you can put anything in here you want as a therapy. And you have to have a promoter that drives the expression of that gene in specific tissues, and then you have to have the gene itself, and then there are a couple other things that we throw in to stabilize the gene, make sure it's expressed well. You can put about 4.7 kilobases of DNA into an adeno-associated virus. So GNE fits into this. GALGD2 fits into this. Other genes are too big to fit into this. So for example, dystrophin is one of the largest genes in the genome. You can't fit the whole gene into this vector. But you can squeeze it down to a smaller version that is also therapeutic. So we just add back these other pieces of DNA and transfect them into a human cell, and it'll spit out a virus where now, instead of having the viral genome, you have your therapeutic gene. And one of the wonderful things about working at Children's, where we have a GMP manufacturing facility to make AAV for patients, is that the technologies that we use to make the virus are actually identical for the clinical production to the production we use to do research experiments. So when I order a virus from the 70 or so people that now work at this facility, they will go through the exact same steps with the exact same reagents that they would to make a clinical grade vector, and then they would hand that to me to do experiments in the lab. And so the FDA loves that because we can actually document exactly the quality of the material every time. And what you get are these little 20 nanometer viral particles, and inside that you have a piece of DNA, and this is an electron micrograph that shows you the packaged DNA inside that viral particle. And because of a lot of years of technology, we can then purify these packaged particles away from these unpackaged particles. We can guarantee that we have at least 90% of the packaged particles in the material. We can run a Western blot or Kamasi gel and look at the proteins in there, and we can see only the 3 proteins that make up the viral capsid. There are absolutely no contaminants, usually 99 or 100% pure. And so you're getting the same thing every time. It makes experiments a lot easier to do. And so point 2 here is that, of course, gene therapy has become very exciting because there are a lot of clinical trials going on right now, over 60 right now just using AAV. And a lot of these are very successful. Perhaps the most successful is the RPE65 gene, which is now called Luxturna, which actually is injected into the retina of patients with RPE65 deficiency, and these patients now can go from not seeing at all to actually seeing. So it's a very dramatic transformation in the nervous system that allows for sight. The most exciting, and then there are hemophilia clinical trials where people are treating the liver with factor 9 or factor 8, and showing that patients with bleeding disorders can have essentially no or very few episodes of bleeding after they receive their gene therapy. The most successful trial at Children's has really been this spinal muscular atrophy trial. This is a gene therapy, again, a gene replacement therapy by Brian Kaspar. It was developed at Children's using an animal model designed by Arthur Burgess, and the clinical trial was done by Jerry Mendel. And SMA1 is a very serious, severe motor neuron disease. Children often die from this disease by the two years of age. And all of the treated patients in this trial lived well beyond that, and even several of them actually have the ability to walk, which is profoundly different than their normal disease progression. And so there are also muscle diseases, I've messed up, like myotubular myopathy and microdystrophin gene therapy for Duchenne muscular dystrophy that are showing similarly encouraging results in some of the clinical trials. And so at our center, we actually have ongoing trials or trials that are about to begin in Duchenne, SMA1, a variety of limb-girdle muscular dystrophies, Charcot-Marie-Tooth disease, several lysosomal storage disorders, mucopolysaccharidosis, and also Batten's disease. So I'm going to first tell you about our journey to develop a clinical gene therapy for Duchenne. And I'm really going to make this very short because I know this is not really probably the thing you're most interested in hearing about, but it just sort of provides an example of how you can go through the process of developing a gene therapy and get it to clinic. So this gene therapy takes advantage of the fact that glycosylation in muscle is actually different in different domains of the muscle. So these long tubular cells here are muscle cells, and where the motor neuron touches the muscle is what we call the neuromuscular junction. That's where communication between the nervous system and the muscle occur. That's how the brain tells the muscle to contract, and that's how you move. And so, no, this isn't working. And so the glycosylation is different. Kelly told you a little bit about lectin staining. This is an example of lectin staining in a muscle where you've used a lectin to a terminal GalNAc called WFA. And you can see here in a single teased muscle fiber that you're only staining this neuromuscular region. So the sugar there is unique and it's different than in the rest of the muscle cell. So we designed a gene therapy where we could convert that synaptic sugar expression to glycosylate the entire muscle membrane with that one sugar. So that is done by a gene called GalGD2, and you can go from having the sugar just in this one little domain of the muscle to now having it be around the entire muscle. So each one of these circles is a muscle cell cutting cross-section. And to make 10 years of work summarized in one slide, this is basically what we discovered, which is that at the neuromuscular synapse, you have this unique enzyme, you have this unique kind of glycosylation, and on this protein called dystroglycan, and it associates with a bunch of other proteins that are unique. But all of those proteins are similar in structure to proteins that are missing in forms of muscular dystrophy. So when we overexpress GalGD2 out here, all of these other proteins are now also expressed out in the rest of the muscle membrane. And by doing that, they can compensate for the loss of these proteins in these various forms of disease. So we first discovered this by making transgenic mice back in 2002, and then I moved to Columbus in 2004, and it took us a couple of years to develop gene therapy and prove that it worked in these 3 different forms of muscular dystrophy. At that point, Jerry Mendel and I went back to the NIH and asked for grants to develop these as products, and that took several years, and then we did a 3-year toxicology study to prove that it was safe, and then filed an IND with the FDA to do various modes of delivery, starting with intramuscular delivery, now with isolated limb infusion, and ultimately now with intravenous delivery. So all of this work was just funded by the National Institutes of Health from NIAMS, the Musculoskeletal Skin and Arthritis Institute. And then this last trial here was funded by Parent Project Muscular Dystrophy. And so even the clinical trial was funded by NIH, and I'm just going to share with you one result. And since I just learned this morning that this was going to be streamed, I'm only going to tell you about data we've already disclosed publicly. So this is data that we showed last year at the Society for Gene Therapy meeting. And here, again, you can see the lectin staining of a muscle in a biopsy taken 4 months after treatment. And this is the biopsy taken in the contralateral leg before treatment. And this is the lectin staining showing this changed glycosylation in the muscle cell. And here, the lamina just shows you where all the muscle cells are. And so we spent a lot of time working on defining the amount of glycosylation, the number of cells we needed to glycosylate, to get a functional change, to define the minimal amount of vector we needed to get that change. And we started our clinical trial with that minimally effective dose. And we got exactly the right amount of expression to get that dose into a patient. And the take home of that is that it looks pretty good. So this boy with Duchenne was over 8.5 years old when he was treated, so he was already quite impaired in his muscle strength measures. But if you look at his leg flexion and extension from pre-treatment and then after treatment, he's actually improved a little bit in most of those measures. And we're seeing even better results with a higher dose in the next patient. So we're certainly stopping the disease from progressing, but we might even be improving it a little bit. So what about G&E myopathy? We got interested in G&E myopathy really through the back door, and we managed to get a grant from the NIH to do a 2-year study looking at evolution. I'm going to explain a little bit about this to you. So sialic acids are also called neuraminic acids or N-acetyl-neuraminic acids. There are really 2 predominant forms in almost all mammals. There's N-acetyl-neuraminic acid and N-glycosyl-neuraminic acid. So Nu5AC or Nu5GC. And about 2 to 3 million years ago, humans got an insertional mutation in their CMAH gene, which made it impossible for us to make Nu5GC. So sometime between the great apes and the evolution of humans, this gene dropped out of our genome. And so what that means is that this one oxygen that this made to make Nu5AC into Nu5GC no longer exists in humans. And it's present in all the other animal models that you would test for disease. So this is just another cartoon to show you that. So basically in a mouse muscle, you have a 50-50 mixture of these 2 sugars for your sialic acid. And in a human, you're missing this one. You're not missing any sialic acid, it's just replaced with all red. And so Ajit Varki, my collaborator on this project, made a mouse with the exact insertional mutation of the human gene into the mouse and created a mouse where you couldn't make this sugar. And this was made a long time ago. And I was interested in what does this do to disease? What does this do to a mouse disease? And so we crossed these CMAH mice into MDX mice, which lack dystrophin. So this is the common model for Duchenne muscular dystrophy. And we're showing staining for N-glycosyl-neuramic acid. Now you're saying to me, at least the scientists are, how on earth are you staining for N-glycosyl-neuramic acid if it's present in all organisms? Well, it turns out birds never evolved this gene. So you can actually make antibodies to this sugar in chickens. And then you can purify it very carefully over glycosyl reagents and not over acetyl reagents, and you can get a very clean antibody. And Ajit Varki developed this antibody, and you can see there's none of this sugar in human muscle or in mouse muscle where we've engineered it. When you cross it into the disease model, you start to see some in regenerating muscles, but not in most of the muscle. And we wrote a science paper about this where we showed that not only did we change the glycosylation situation for sialic acid to the more human-like condition, but we saw increased inflammation, increased weakness and wasting, and increased death that was more akin to what boys with Duchenne have than what mice with missing dystrophin have. And so we convinced the NIH that, we should do this for G&E myopathy because it's a disease where sialic acid is affected. And so you've seen this slide before, but G&E affects the committed steps in sialic acid biosynthesis, taking UDP-glucnac and converting it to MANAC6-phosphate. But later down in the pathway, you have CMAH converting Nu5AC to Nu5GC. So we decided, we wrote to the Nishino Noguchi group and asked them if we could have their disease model for for G&E myopathy. And they were very, very generous and mailed us these mice from Japan. And so you've heard this before, but these mice are small, their muscles are small, they develop muscle weakness, rimmed vacuoles, inclusion bodies, and have increased death. And so we thought this would be a great model to test. And we crossed it to the CMAH mice. So now we have a double knockout transgenic mouse. So it's a sort of a complicated genetic experiment. And then we asked, what happened? Well, what happens to the sugar, and this is staining for N-glycosyl-neuramic acid, or Nu5GC. You can see that there's less of this in the mouse model around the muscles. And then, of course, in the CMH deficient muscles, it's gone. So that worked. But what we discovered was that the phenotypes that were described in the original paper had drifted dangerously more towards normal. And this is not an uncommon thing in mouse biology. If you're making a mouse model for a muscle disease, now over 60 generations, you're breeding it, you tend to select for healthy mice that are breeding more successfully. And so especially in a transgenic line, you can then breed out the disease to some extent by doing that over time. And so this isn't to say that the mice have were not reported correctly, but by the time we got them 10 years after this, they really didn't show any death, and they didn't show any change in weight, except the females showed a little change in weight, but the knockout was no different than the single mutant mouse. The muscles were no different in weight, but the mice were sick. They developed enlarged hearts, and they developed massively enlarged spleens. So there's something about G&E that's wrong in the mice, but it's not really muscle specific. We looked at muscle strength, really no change. We looked at walking, really no change. So that told us that we kind of couldn't do the experiment we wanted to do, which is to test disease severity. But Kelly was in the lab, and she needed to get a PhD. Here's the muscle pathology. This is Congo red staining to look for inclusions. Here's a positive control. This is a brain from an Alzheimer's disease mouse where you can see the red spots, but we just didn't ever see inclusions in these muscles. So what we decided to do was look at sialic acid as a therapy, because N-glycosyl acid is made by this one gene from NU5AC, and it never goes back. There's no gene to put it back. So once you put it in, it has to stay as GC. So we decided we could look at this as a way to look at how much sialic acid actually went into different tissues when you give it orally, when you give it as food, when you give it as gene therapy, and then ask about actually be able to visualize where the sugar went in the cell. And so this is an example of this experiment. This is an oral sialic acid experiment where we added 2 mg per kg of sialic acid. And Kelly did this by oral gavage because we wanted to make sure the mice got the exact amount of the sugar. And then we waited 1 week and we asked, where does this sugar go in the body? Does it go to muscle? Does it get on the membrane where it's supposed to be? These sorts of questions. And I don't know if you can see this, but these red lines here are staining for sialic acid. And so when we gavage with NU5AC, of course, there is no GC, and so that's just sort of your negative control. And you can see that actually it goes in, not only goes into muscle cells, but it gets out to the membrane, and it gets expressed along the membrane in a very productive way. But not in every cell, and not every muscle is the same. So the gastrocnemius, the large fibers, the type 2 fibers that do the higher impact work tended not to be positive. In other muscles like the quadriceps, they tended to be positive. So these are all leg muscles, and then the tricep is an arm muscle, the diaphragm is your breathing muscle, and then this is the heart. So the diaphragm did really well at taking up the sugar and putting it out on the membrane. The heart did a good job of taking up the sugar, but you don't see any lines here, and that's because there actually is no expression along the cardiac membranes. So the heart actually doesn't get it out to the surface in a very useful way. We looked at all the organs, of course, and so the stomach is where the sugar is going in. It's very, very bright. The kidney showed a very variable pattern where some tubules were expressed and some were not. Lung was much more even. But the striking thing to me was that the liver was actually there was no sialic acid in the liver. Unlike muscle, we didn't really load any sugar onto the membranes of hepatocytes in the liver. And I'm going to come back to that with the gene therapy in just a second. So Kelly did a lot of these variations in this experiment. She asked about food. So we actually created our own mouse chow where we enriched the protein source as porcine submaxillary mucin. And this creates 40 times the amount of glycosyl of normal mouse chow, basically like a 10 hamburger a day diet for a mouse. I'm not going to show you any pictures because actually we didn't load any sialic acid into any muscles using a dietary protein source as our source of the sugar. We could see it in the colon, in the intestine, where the food goes into the body, but we didn't see it get to other places. So what about gene therapy? I'm going to tell you two very short stories about this. So we took the normal allele for the 722 amino acid G&E gene from humans, and we made AAV vectors. So this tells you the serotype of the vector, and at Children's we like to use this serotype called Rhesus 74 because it was discovered at Children's. And it's very good at going from the blood into tissues. So you give this gene therapy via the blood, and it manages to go into muscle and heart very well. And we use a muscle-specific promoter. And then we also tried designing these with a liver-specific promoter and also a constitutive promoter. I'm not going to talk about that data. And then we delivered directly to the muscle by injecting it, or we delivered intraperitoneally to deliver it more or less directly to the liver. So this just is an experiment where we stained for G&E made by the gene therapy. So the G&E that we put into the cells had an epitope tag on it, which is a little bit of extra protein that you can then see as being different from the G&E that's already in the animal. And so you can just see what you made from the gene therapy this way, and you see this bright red cytoplasmic staining with normal G&E or wild-type G&E, and also with the D to V mutation and the M to T mutation that cause G&E myopathy. And the take-home message here is you can overexpress G&E protein at very high levels in the muscle, and it goes right to where it should normally be, and it doesn't get, there's no problem with that. Likewise, there's no problem with the mutants either in terms of their expression in the muscle. However, because we had this epitope tag, we could actually precipitate the G&E out of the muscle and then ask about it as a protein. This is a Western blot to show you that. So what we're showing here is the G&E protein at 1, 3, or 6 months after injection. And this is what the wild-type protein looks like. This is what the D to V mutation looks like. And this is what the M to T mutation looks like. And there are a couple take-homes from this. One is that obviously we could produce a lot of G&E protein using gene therapy in the muscle, and that it grew over time. This is from a single injection at time 0. So at 1 month, you have expression, and we expected that. At 3 and 6 months, you have even more expression. The two mutations that are specific to G&E myopathy show much less expression early, but then eventually it catches up and you get normal amounts of expression. What was intriguing to us is this multimer up here. So this is kind of like, so proteins exist in three-dimensional shapes, but they also bind to each other as single units to make what we call quaternary structures. So it's kind of like building a snowman. You have at least in the Martin family, we have 3 parts to a snowman. We have the base, the middle, and the head, right? And so G&E sort of works that way. Here's the base, a single subunit, but up here you have all 3 subunits bound together, which is generally the form that's thought to be enzymatically more active. But if you look at the D to V or M to T mutations, you really see much less of that upper level snowman. And so we think that actually in muscle, this might be a sign that we could explore for how G&E is different when the mutations happen. That's tissue specific to muscle that changes how it behaves in the cell. And we're hopefully going to continue to look at that. The other thing we looked at, of course, is the sugar. And so this is an uninjected muscle and an uninjected liver. If you put muscle-specific gene therapy into a muscle, you can see these circles become brighter. And so you're getting increased glycosylation of the muscle. You're obviously not getting increased glycosylation of the liver. But if you put a liver-specific promoter into the tissue, you get massively increased glycosylation in the liver, but you actually also get even more glycosylation in the muscle than when you use a muscle-specific promoter. So the liver actually secretes glycoproteins into the serum. It's the main source of protein for the serum. And so we actually think that this liver-specific expression of GNE is actually populating sialic acid into the muscle in a way that may or may not contribute to glycosylation in terms of function. And so that's just something to keep in mind when you're designing these therapies is, it may be that the affected tissue is the only tissue you want to treat, but it may be that you want to treat other tissues because they contribute to glycosylation of tissues beyond the muscle. I'm just going to show you one experiment that Kelly didn't want me to show you because she didn't do it very many times, where we started to address this question, can we make a better model for the disease? And what she did was she made these microRNAs. And what these are, like the word micro suggests, they're little tiny RNAs that have a sequence to GNE that allows it to bind to the normal transcript and degrade it. So it's a way to reduce the gene expression for GNE. And Kelly just did some experiments in cells and found that this construct, MI4, worked the best. And then we put in a second gene into an AAV vector, which was green fluorescent protein, so we could see where this microRNA went, and we injected it into muscle. And you can see this massive green fluorescence because we've saturated the entire muscle with this construct. And then she showed that if you inject this into a heterozygous GNE mouse, so this mouse has a little more than half of the expression of a normal mouse, but if you go over time out to 6 months, you can actually knock it down from about 60% of normal to about 35 to 40% of normal. If we did that, we actually started to develop and see inclusion bodies in certain muscle fibers. So this is just a Congo red stain by refringence, rhodamine fluorescence. This all just tells you that that's an inclusion. And what that tells us is that by reducing GNE, we can actually maybe make a better model for the disease. And that's sort of what we're aimed at doing right now. So the take homes from this study, which is I'll give you as sort of a modest study, is that we need a new mouse model to study gene therapies, and we're working on that right now. And that we think that reducing gene expression will be sufficient to develop that model in the absence of any known GNE mutation. That GNE mutations may alter the quaternary structure of the protein, and that the good news here is that GNE protein overexpression in muscle can be tolerated at very high levels. So there may not be an upper window to where you can work for getting the medicine into the patient. And also this caveat that overexpressing GNE in the liver actually leads to increased glycosylation from secreted proteins in the muscle extracellular matrix, which may be an important contributor to changing sialic acid with this therapy. So that's my talk. Thank you very much, and I'm happy to answer any questions you might have.
Thank you so much, Dr. Martin. That was extraordinarily interesting. Do we have anybody with specific questions for Dr. Martin? We have a question over here.
Hi.
Hi.
So I'm not a doctor or anything, and I'm trying to remember as best as I can, but when you were talking about the gene therapy for Duchenne's, and you spoke about the possibility that some of those patients actually improved with the therapy. For GNE myopathy patients, obviously our biggest setback is the loss of muscle. So from what I understand from my MRIs, some of my muscle has actually turned to fat. So when talking about gene therapy, you can make a treatment or slow the progression, but as far as a cure, you can't really rebuild muscle that has already turned to fat. So in those gene therapies, we'd be hoping to improve or reverse the symptoms of what muscle we have left. Does that make sense?
So there's sort of two schools of thought about treatment, right? Most of these treatments are what we would call preventative treatments. They're going to stop the disease from progressing. Certainly gene replacement, that's the idea. There are other therapies like follistatin gene therapy, which is a myostatin inhibitor, that are designed to build muscle. Generally speaking, they build muscle from muscle, not from fat. There's very little data on how to convert fat back into muscle. There are studies on certain proteins that can affect stem cell choices about whether they go down the fat or the muscle axis of decision making. But that is not well developed. But yes, I mean, I think that those other therapies about muscle building, in theory, could be used in concert with prevention to build increased strength. And some of those can be quite dramatic, right? I mean, certainly cattle breeders have figured this out long ago, right? So a lot of cows are bred as myostatin single knockouts, and they look big. There are patient case studies where myostatin is knocked out, and you have these 4-year-olds that look like Arnold Schwarzenegger. I mean, so you can double the amount of muscle with those therapies, double it. So that doesn't make the muscle healthier, but it makes twice as much muscle means you have twice as much strength. So, that's not bad. But converting one tissue to another type is an entirely different problem. Yeah.
So that tends to be a question that we get often, just to make you feel good. And in order to answer that question, not more than turning fat into muscle, can the remaining muscle left in a typical patient be improved upon, I guess, is the way to ask that question, in the case of gene therapy, where we're not just slowing the progression, if we are even to see mild results, is that something that can happen?
That you would have increased strength?
That you would have increased strength or reversal from working from the existing muscle, whatever's left, as opposed to turning the fat into muscle.
Absolutely, absolutely. And I think, I suspect that will be the case for many muscles in G&E myopathy where they're not yet gone, because muscle has an incredible resilience in terms of restructuring itself to become functional again. And certainly in the myopathies where muscle damage isn't the primary cause, it's sort of an internal problem of the muscle, like myotubular myopathy, you see some very significant improvements in function as a result of just correcting the gene.
Right. So as long as someone hasn't completely lost all of their muscles.
Yes.
Yeah, that would be my backup question. My backup question would be, is there, as long as there's tiny little bit of muscle left, is that enough to show improvement? Or is there a point where our muscles have gotten so weak that improvement would not be anticipated at all?
Oh, I don't think, it depends on what your task is. Muscles are designed to lift weight. And so if you have a small weight, then you don't need much muscle. If you have a big weight, you do need a lot of muscle. So it really just depends on the task at hand, whether that would be, but I think as long as you have muscle, yeah, you should have the ability to it should work. Yeah.
Yes, as I say, that's a question we get very, very often, Dr. Martin. That's a question that everybody has. And in terms of, just for clarification, Duchenne, you may not realize, all of these diseases that are being referred to are in fact systemic muscle diseases, right? So they're very similar in that regard to G&E myopathy. So can we expect to see the replication of the similar results, provided everything else goes as planned?
Yeah, I would hope so. I would hope so. And muscle is an incredibly resilient tissue because it has stem cells that can divide and actually make new muscle. And so even if you're missing muscle fibers, but your stem cells are still there, in theory, you might be able to then regenerate muscle where it wasn't before to some level. I can't say exactly how that would work, but it's theoretically certainly possible.